All right, so our topic is water. So, why do plants need water? Well, water is super critical. Water is a limiting resource, right? If plants don't have enough of water, they're not going to be able to perform a lot of these functions like photosynthesis. We saw how water was needed in photosynthesis, right? It donates that electron to replace the one that got excited, right? A lot of different cellular functions rely on water. Right? We rely on water to transport minerals and nutrients throughout the plant, right? In that xylem and in the phloem. We can help support the plant. So water actually gives plants some structural support. And we'll talk about this. It's support in the form of turgor pressure. And finally, water is limiting, but it's also going to be lost from the plant. Right? It's going to be lost through a process called transpiration, which we will also elaborate on today. Right? I like this graph here because it shows you how areas of the world where water is very limiting, right? We see a decrease in productivity here, but essentially the amount of plant growth. Right, so areas that have very little low annual precipitation, we see very low amounts of growth, right? Versus somewhere like a rainforest, right, where that gets a lot of precipitation, right, we're going to see a lot of growth, right? So water really is a limiting resource. Oh, the clicker might not be working today. All right, well, why water? Right? Why are we relying on water rather than some other liquid or substance, right? Well, water has these properties associated with them that make it really ideal as a resource, right? So its chemical properties, the way it interacts with other surfaces, right, really make it an ideal medium, right? And so some of those properties of water, and I actually have a video, Amoeba Sisters, if anyone watches Amoeba Sisters, to kind of walk us through these properties because we're diving into a little bit more of a chemistry for this here, right? But essentially water is a molecule that is polar, right? Meaning the molecule itself has a side that has a partial positive charge and a partial negative charge. Right? It has both charges on our molecule, right? And it's this polarity that gives it these really unique properties, like will be explained a little bit further in the video here. But it's an excellent solvent, meaning things can dissolve in water pretty easily, right? And it can dissolve a lot of different substances. It has a high specific heat, right? So it takes a lot of energy to say boil water, right? Which is great because on a hot summer day, you don't want the water boiling off of our legs. Right? It has these properties of cohesive and being cohesive and adhesive, which is really what we're going to focus on today when we talk about movement of water. Right? How water molecules can stick to themselves, but also stick to other surfaces. Right? And they have a high surface tension, right? So that means it's really hard to break apart the water molecules, right? Some definitions that I think might be helpful as we're watching this video here. Again, I'm trying to simplify a lot of chemistry principles here, right? But we have some bonds happening between the atoms in our water molecule, right? The bonds in the water molecule itself, right? So we have 2 hydrogen, right, and 1 oxygen. They are bonded together via a covalent bond, which means they are sharing their electrons. So those electrons orbiting their nucleus, right, they're actually sharing those electrons. Right, causes them to bind together in that covalent bond. Right. Like I mentioned, water is a polar molecule, right? Which means when they share those electrons, there's actually one molecule that kind of hogs the electron a little bit closer to it, right? That would be oxygen. Oxygen kind of hogs those electrons, brings it closer to the nucleus of the oxygen rather than the nucleus of those hydrogens, which causes this molecule to have a partial positive and a partial negative charge. Right? So there's a charge associated with their molecule. And finally, they can form hydrogen bonds. Right? So a hydrogen bond is this force between a hydrogen atom, right, and a slightly charged molecule, right? Water is a slightly charged molecule. Water also has hydrogen, right? So to bond the molecules of water together would be these hydrogen bonds, right? So within a molecule, they share their electrons in these covalent bonds between different molecules of water, right? They are attracted together via these hydrogen bonds. Right? I simplified quite a bit of chemistry in this one slide here, but I'm hoping that it helps you understand the concepts a little bit better. All right, I do like the visuals there. I think the visuals might help you remember some of these concepts, right? So they went over quite a few concepts and even elaborated on the density of water and how water is less dense as ice, right? One thing I would add to that, if we're thinking specifically of plants, right? So she talked about the pond in her backyard, right? The water would expand when it freezes and create that layer of ice that floats. While in plant cells, right? Plant cells have a lot of water in their vacuoles. When those plant cells freeze and that water freezes in those vacuoles, it actually expands like water does when it freezes, but it expands so much that these cells will burst and die, right? So that's why a lot of times if these plants are exposed to cold temperatures, right, after they're exposed, you might see that their leaves starting to die, right? I've done that to plenty of my plants, but it's because of the water expanding and freezing and bursting these cells. All right, a couple of those properties that I really want to focus on, though, are cohesion and adhesion, because they play such a huge role in drawing water up through the plant, right? So cohesion is the attraction of water molecules to each other, right, through those hydrogen bonds. And adhesion is the attraction of water to other surfaces, right? So like the walls of the xylem, right? If you've ever poured a glass of water, right, and it starts pouring on the side, starts to make a huge mess, doesn't pour very nicely, right? It's because of those adhesive forces that water's trying to stick to the glass instead of pouring nicely in that stream, right? So that's a common example that many of us have experienced, right? And then if we think of a plant, right, like I said, this cohesion and adhesion really plays a role in bringing water from the roots all the way up to the leaves, right? We'll end up breaking down this image step by step here, right? But cohesion and adhesion, two very important properties out of a list of very important properties of water. All right, so properties of water. How do they function in pillows so that this water is not going to move around? It kind of stays in place. All right, so let's do a couple of discussion questions after all of those videos. Maybe get up and stretch if you need it here. I want you to think about how might the properties of water change if water was not a polar molecule, right? Give it your best go here. And what properties are causing that water molecule to form a sphere in space, which we kind of talked about.
These questions are like so different. Yeah, like these hydrogen oxygen would stay together. But like, it's actually ridiculous to send out the questions. Like these are kind of hard. Like, how am I supposed to shit on it? Or is it like oxygen? Okay, do you think I should ask you? I was thinking about the surface tension in the sphere. So like
And then the properties that cause miracles. It's the adhesion. Or no, wait. Yes, cohesion. Oh,
I like it.
and
Yeah.
cohesion.
Yeah. I tried your own. If you're wondering, I guess not water. I have not checked yet this morning.
I checked this at like 10 o'clock.
Yeah.
I love when
Okay. All right. Yeah, campus safety is doing more now. I don't know. I want it very early.
Oh, I guess not.
Alright. I like sometimes how the captions stop on these like really random misinterpreted lines. Don't know, I think I said something about cohesion and it said like his mom's responsibility. I don't know. It makes me laugh sometimes. Alright, so thought questions. How might the properties of water change if it was not a polar molecule or it was non-polar? What do we think?
It'd be like a gas like methane, I think, so would it stick together because it doesn't have the opposites?
Yeah, it wouldn't stick together as well. Maybe it has a lower specific heat, which means it might boil and turn into gas quicker. Yeah, what else might happen?
It's more of a question, but does that mean that the oxygen would stay away from it? Is the polarity say, because like it's saying that it hogs the oxygen, so does that mean that it doesn't care about oxygen anymore? And it's just gonna like,
It would still care about oxygen, so oxygen is hogging the electrons
Yeah.
from the hydrogen, and if it's not polar, it wouldn't be hogging those electrons anymore. They'd be shared very equally. So they could still be in these bonds, but they just wouldn't be polar. Yeah, yeah. What else might change if it wasn't a polar molecule? What other properties does it have? Would it likely form a sphere in space? No, because our second question, what property is causing the water to form that sphere in space? What was that? Yeah, surface tension and cohesion, right? Those hydrogen bonds interacting. Right? If our water molecule was not polar, we would not be able to have hydrogen bonds, right? Hydrogen bonds rely on polarity of molecules, right? So essentially, every property we have talked about would no longer be able to exist if water was not polar, right? So it's very crucial that water is in this polar molecule and has that partially negative and partially positive charge associated. All right, let's start talking about how to move water, all right? So there's a couple of different ways that water is going to need to move in a plant, all right? The first way we're going to talk about is short distance transport, right? So rather than transporting water from the roots to the leaves, short distance transport is like transporting water between two adjacent cells, right? Or in very close proximity to each other, right? In order to do this short distance transport, essentially the substances are going to cross the plasma membrane of our cell, all right? So if we think about our cell here, right, it's surrounded by this cell wall, which is like this mesh of different molecules, right? Water can easily pass through that mesh of different water molecules. But then it has to pass through this plasma membrane to get inside or outside of a cell, right? So this plasma membrane, we would say it's semi-permeable and selective, which means it does not just allow anything to pass through it, right? It's selective in what is actually going to be able to move through it, right? Now, we'll talk about water and how water moves past this plasma membrane here, right? A lot of different solutes, right, different molecules, things that have been dissolved in water, can't get through this plasma membrane. In fact, have to use some proteins to help it along, right? We'll talk briefly about that as well. All right, so we're talking about transport across a plasma membrane, so into or out of a cell, right? One of the ways that transport occurs is this type of transport called passive transport, right? So passive transport is going to move molecules from areas of high concentration to low concentration, right? So from areas where these molecules are very abundant to areas where these molecules are not as abundant, right? We're always going to move from high to low in passive transport, right? In passive transport, right, the cell doesn't have to use any energy, right? So we don't have to use up any of our precious ATP molecules to make this happen, right? There are 3 types of transport that we will briefly talk about, right, because they are important when we talk about water movement, right? The first is osmosis, right? Then we'll talk about simple diffusion and facilitated diffusion. Right? Let's talk about osmosis first. All right, so osmosis is the movement specifically of water, right, along a gradient, right? So when we think about the movement of water along this gradient, right, it kind of relies on the amount of solutes that are present, right? So I like this image because it gives a really nice visual here. So we can imagine, right, we have this beaker and we have this semi-permeable membrane in here, right? Something that's very similar to a plasma membrane. It only allows certain things to move across it, right? We also have this water, right? We can see the water is kind of indicated by this lighter shade of blue and also these smaller blue dots, right? And we have these sugar molecules dissolved in the water, represented by these larger pink dots, right? Now, if we look at kind of the ratio of water to sugar, right, we can see on one side of this membrane, right, on the left-hand side here, that there's way more water molecules per sugar molecule, right? Now, we want to reach kind of like an equilibrium. We want it to have this equal amount of water molecules to sugar molecules, right? So what's going to happen is on this other side here, where we have way more sugar molecules, and I say way more, means we have 3 more in this image here, right? But we still have the same amount of water, right? Now that's a very different ratio, right? There's slightly more sugar molecules to water molecules. So we want to kind of balance that out. And the way that this is going to work is that water is going to be able to move through this semi-permeable membrane, but sugar will not be able to move, right? So only water can move, and water is actually going to move towards the side with more of these dissolved substances, so in our case sugars here, in order to kind of equalize the amount of water molecules to sugar molecules, right? So when we're thinking about osmosis, we have to think about the dissolved substances. Right? If there are more dissolved substances on one side of a membrane, water is going to end up moving towards that side with more of those dissolved molecules. All right. So osmosis really deals with the movement of water with a solute present, right? Something dissolved in that water. But we can also say that water can move with simple diffusion, right? Meaning water can simply diffuse or move across the plasma membrane, right? And again, it's moving according to a gradient, right? This time we're not paying attention to the solutes. We're just looking at the amount of water molecules. And we can see that we have a lot of water molecules on the top of our plasma membrane and very few on the bottom. And so we're going to move these water molecules from concentration of high to a concentration of low water molecules. And so they're going to simply diffuse across this membrane, right, until both sides are relatively equal in the number of molecules present, right? So this would be simple diffusion, right? So osmosis, water is moving across a membrane to kind of equalize the amount of water molecules to something that's dissolved. Simple diffusion, right? We are moving molecules across a gradient from areas of high to areas of low of that specific molecule. Right? But there's one other way that water can move, right? And that's through a process called facilitated diffusion, right? So this time we're still moving our molecules from an area of high concentration to an area of low concentration, right? But it's being facilitated, it's being helped by some sort of a protein, right? So we would have a transport protein, so a protein that spans the width of our, maybe I should say the height of our plasma membrane, right? It crosses that plasma membrane entirely and basically acts like a channel or a pore or an opening in this plasma membrane that these molecules can go through. Right? For some molecules, right, larger molecules, maybe molecules that have a like a higher charge associated with it, right? They actually need to rely on these proteins. They can't simply diffuse past the plasma membrane layer, right? Water can simply diffuse, but water can also use one of these channels, right? And water specifically uses a channel called aquaporins, right? So it's a channel very specific to moving water, right? That's all it does is move water, right? And these aquaporins play a major role in water uptake from a plant, right? If we're trying to move water across a plasma membrane, it's a relatively slow process. It occurs, but it's slower, right? But if we add in these channels, these aquaporins, all of a sudden we can increase the rate of some different solutes here, and they are dissolved in water. I want you to tell me which way the water would move, right? And then if this was a real plasma membrane, what could increase our rate of osmosis? So a little bit of a review here. Oop.
I think you said the one side.
Yes. Yeah.
So if it's always happening. So if like these things are always does it ever stop? Because it like water transfer
So once there's equilibrium, there's still going to be movement, but it's at basically the same rate on either side. So even though there's movement, we're still maintaining equilibrium. Now, if we were to reduce other solutes or molecules, then that might change which way the water goes. Yeah.
Oh.
No,
Pretty
always.
sure.
Yeah.
It's not you.
The way I looked over to
I
you.
know. $7.20.
All right, we have our beaker filled with water and dissolved solutes here. Which way is our water going to move? To the right or to the left?
the right.
To the right, yeah. Why is it moving to the right?
To dilute the solutes that are on the right, so that way eventually they're equal.
Yeah, to try to get to this equilibrium where there's equal amounts of water to solute ratios, right? So this water here would go to the right, and eventually we would see the water level here be much higher, right? Trying to dilute, I think that's a great word there, dilute the amount of solutes. All right, if this was a real plasma membrane, what could we incorporate into this membrane to make this process even faster? Aquaporins, yes. Our proteins, they span the length of our plasma membrane here, and they allow for faster movement of just water molecules. Any questions? All right, we're going to go into the next principle, and I think after the next thought question, we'll take a little break here. Oh, I keep trying to use my clicker. I forgot. I think it must be dead. All right, so we talked about transport across plasma membranes, right? Water can move in and out of cells due to osmosis, right? So we're really looking at those solute concentrations on either side of that plasma membrane, and that's dictating which way the water will move, right? We can dive a little deeper into this concept and talk about water potential, right? So I believe one of the videos I had you watch talked about water potential here, right? It's an equation that takes into account solutes, right, as well as some other processes that dictate the way that water will flow. All right, so water potential. It's this hugely important concept in plant biology, right? It's going to tell us which way is water is going to flow, right? And it's going to be measured as a megapascal, right? So you might see this MPa after some of our numbers here. That refers to our megapascals. But it's basically the measurement of free energy of water, right? It tells us the ability of water to do work, right? Or to move, essentially, in a given volume of liquid, all right? This equation is influenced by a few things, right? First of which is the solute concentration, right? Which we've already talked about and mentioned here, right? So we would say that the water potential, right, takes into account this solute potential. So it's this Greek symbol, which I forget the name of this Greek symbol. Is it psi? Do I have anybody that knows the Greek alphabet? I think it's psi. Right, but this Greek symbol with subscript S, so that's solute potential. With our, it also takes into account the pressure potential. Right, we'll elaborate on that. So the subscript P, and it also takes into account the gravity potential. Right, so it's taking into account all these different factors to determine how the water is going to move. Right. Now, water potential is something that's always going to be talked about in reference to another water potential, right? If I said the water potential was, let's say water potential was 5, right? That's absolutely meaningless unless I'm comparing it to another water potential, right? If I say the water potential is 5 inside of this cell, and the water potential is 3 outside of that cell, now that's going to tell me something about which way the water will move, right? We'll get into that. All right, water potential, right? We're always going to look at a number, right? We're going to have a number in megapascals here. And when we think about water potential, water is always going to move from areas of a high water potential to an area of a low water potential, right? So if inside of the cell has a higher number of a water potential, say it's 5, right? And outside of the cell has a lower number, say it's 3 megapascals, water is going to move from the area of high to low. So in that case, the water would move outside of the cell, right? So this is kind of like the mantra of water potential, right? If you remember this, right, that is fantastic. Water is moving from high to low. All right, so let's talk about our water potential equation and all the little steps that make it up. First of all, right, I said it's a value of the solute potential, the pressure potential, and the gravity potential. Most of the time we ignore the gravity potential, right? That typically doesn't come into play unless we're talking about trees or plants that are really tall, right? So oftentimes it's ignored. We're still not going to talk about it, even though, gravity plays this role in space, right? We're still talking about plants that are relatively short growing in space. We're not talking about trees that have to go against gravity, right? And I'm not even sure that research has been done on transpiration and gravity in space yet. Right? So we're going to ignore gravity. So for the purpose of this class, we could say water potential is the solute potential plus the pressure potential. All right. If we have a beaker of pure water, right, there is absolutely nothing dissolved in this water, right? It is sitting at room temperature, right? It is at sea level, right? So if we're thinking about pressure, it's the pressure exerted on it at sea level, right? We would say that this pure water, right, in an open container at room temperature at sea level has a water potential of 0 megapascals, right? So that's kind of our base point here, right? This is almost never going to be the case, right? You'd have to work really hard to get pure water, first of all. Second of all, you're never going to find pure water in any organism, right? There's always going to be something dissolved in that water, right? So this is where we have to start adding in all these different influences to our equation, right? The first being the solute potential, right? This is the effect of anything that's dissolved in that water, right? So in plants, right, if we think about the solutes in plants, things that are dissolved in water, those are going to be minerals and nutrients. They're going to be sugars that the plants have made. They might be hormones, right? Now the solute potential of pure water is 0 because there's nothing dissolved in it, right? But like I said, that's almost never going to be the case. There's always going to be something dissolved in water inside of an organism, right? And so when water binds to any of these solutes, right, it decreases the amount of energy that it can do to do work, essentially, right? So if water is tied up trying to dissolve something, right, that means it's lowering its potential energy to do anything else, right? And thus the solute potential is always going to be a negative value, right? So when we're looking at our equation, that solute potential is always going to be a negative number because it's decreasing the amount of work that a water molecule can do because it's already tied up and doing something else, right? So if we look at this solution on the bottom here, right, now we have some dissolved sugars in this solution, right? And if we look at the solute potential here, we see the solute potential is negative 0.244 megapascals. Right? So in an open container of water at sea level where we still don't have the influence of pressure, right, we would say the water potential is the pressure potential of 0 plus the solute potential of negative 0.244 megapascals, which means the water potential is negative 0.244 megapascals for the water potential. Right? So solute potential is always going to be a negative value. All right, the other half of our equation here is the pressure potential, right? So this is the physical pressure that's exerted on a volume of liquid, right? Now this could be a negative or a positive value depending on the pressure that is applied, right? We could have negative pressure. So if you think about water in this syringe here, if you were to put your thumb over the opening of the syringe and pull that syringe back, right, it's like this pull on those water molecules, that would be a negative pressure, right? Negative pressure is like a pull, right? So it would be a negative pressure, and we would see a negative pressure value, right? Very different if we had that same syringe, right? Again, we put our thumb over the end, but this time we try to push that plunger in, right? That's a push, right? It's a positive pressure being exerted, right? In this case, pressure would be a positive value, right? So we could have negative or positive pressure. Now in plants, right, we're mostly going to see negative pressure because water is being pulled up through the plant. All right, so kind of alongside pressure, right, we also have turgor pressure, right? Let's see, would it be not necessarily with the water potential equation, but it's still important to mention when we talk about pressure in a plant, right? So turgor pressure is the force generated from the plasma membrane pushing against the cell wall. So the cell wall is always rigid, right? It stays put, right? It's going to be in that boxy shape, right? But the plasma membrane can have some wiggle room. It can contract a little bit, it can expand, right? And so when we have water in our vacuoles, right, and our vacuoles are filled with water, it's going to be pushing that plasma membrane out against the cell wall, right? So this is creating a lot of turgor pressure, right? This is the type of pressure we want to see in our cells, right? This keeps our cells, we would call it turgid, right? It's going to be upright, it's going to be full of water, it's going to look like a healthy plant. Now on the other hand, right, if we don't have enough water in those vacuoles, that plasma membrane is going to start to shrink away from our cell wall here, right? And when this happens, we get what's called a flaccid cell, or we could say a flaccid plant, right? This would be a very unhappy plant. This would be a plant that is starting to wilt from lack of water, right? So turgor pressure, important to mention when we're talking about pressure in a plant, right? But kind of a side thought to water potential. Any questions on turgor pressure? All right, let's put this water potential equation kind of into practice here and run through a couple of different examples so we understand how it works. All right, so we're going to go through this example where we have this solution here, right? This solution has some sucrose, some sugars dissolved in it, right? And because it has some solutes, right, we would say the water potential, right, is the pressure potential of 0 plus that solute potential of negative 0.244. So our water potential of our solution is negative 0.244 megapascals. Right? Now we're going to take a cell, right, this flaccid cell, right, and drop it into that liquid. Now this flaccid cell, right, again doesn't have the influence of pressure right now, right, has a solute potential of negative 0.732 megapascals, which gives it an overall water potential of negative 0.732 megapascals, right? We're going to drop this cell into our sucrose solution, right? And when we do so, we see that the water is moving from an area of high to an area of low. Now, the negative values can start to mess with your mind here, right? So this solution, right, is negative 0.244 megapascals, which is a higher value than the negative 0.732 megapascals, right? So because this is a higher value, even though it's still in the negatives, it's still a higher value, all right? Water is going to move from areas of high to areas of low. Water will flow into that cell, right? Oh, I guess I had some more transitions in here. So paying attention to the numbers of our water potential, right? Water is flowing from areas of high to areas of low. right? So when this cell has enough water inside of it, right, it went from a very flaccid cell where that plasma membrane is pulling away from our cell wall to a very turgid cell. It now has a lot of turgor pressure. All right, so we're going to take that cell that has now become turgid, right, from putting it into that initial sucrose solution. Right? It now has a water potential, right, of 0.244, negative 0.244 megapascals. Right? We're going to drop it into a sucrose solution, right, that's more concentrated. We have more sugar inside of it, more solutes, which means more solutes, we're going to end up having a more negative value. And we can see that reflected here, right? We have a more negative value of our water potential of negative 0.732 megapascals, right? So we're dropping that cell into our water. And this time, again, applying the same principle from areas of high to areas of low, water is going to flow out, right? Because outside of the cell has a much lower value than inside of our cell, right? So our cell will then become flaccid as the water leaves. All right, so I mentioned water potential is hugely important in plant biology, right? It really is such a big deal, right? Not only does it determine which way water is going to move, right, but it can indicate how well watered the plant is. So if we think about the whole plant, whether it's turgid, whether it's flaccid and wilted, right? Water is used in a lot of different plant functionings, like photosynthesis, right? All these other different functions, right? Making proteins, expanding cells, right? Opening and closing our stomata, right? We can see in this graph here all these different physiological changes that can happen when this water potential becomes really low, right? So if we have pure water at -0 here, which is hardly ever the case, well-watered plants typically exist within this -0 to -1 range. On the opposite end of the spectrum, we have plants in very arid desert climates where the water potentials might be like -4. And we can see the more negative we get in our water potential, these different functions start to peter out or not be able to happen at all, right? So this is why determining water potential can be hugely, hugely important. All right, let's do a thought question, and I'll give you a chance to practice these concepts here. All right, so I have given you a cell to the right here with a water potential of -0.364 and a couple of different scenarios. Right? I want you to apply those scenarios to our cell to determine which way water is going to move. How's it going over here? I know you'll be gone tomorrow. You're missing the greenhouse activity.
But like, I just
Want to
don't
do it
want
in class
to
today?
think there's
Do you have time? That works?
Like trainers upstairs.
Yeah. Yeah. All right.
Why does it have such a good CF value that like we can't have the CC? Because that's what happens with my CF. Like, oh wow, That was really good. You did really good with the troublesome football players. That's why you guys was.
Not tomorrow.
I was so excited.
Water potential, how's it going?
Good.
It's going.
This definitely would have been a really good class to take before. Like
Yeah.
if you did 101 this class, then 102. Because I remember taking 102 with a girl that took Astronaut, and she's like, oh yeah, we like talk about this class. We have
I think I know who you're talking about. Yes.
So,
Go through a lot of the same concepts. Yeah. How's it going? What's our point of view?
Whether it stays or goes, if it's higher. So the negative.
I think most of the games are,
Yes.
except for me.
It's closer to that zero, right? And zero being the high number.
The water goes from high to low.
Or it's a BT.
Yeah, it's a little different.
Which way?
Out. Exit.
Okay, so we did it.
Yeah.
You would not believe how long
Except for
it took
Cleo.
to wrap my head around my thing.
Let's go.
Allowing for us to introduce this messy. It's so bad.
Okay, so then the second one, it's gonna enter.
All right, we have our cell up here, right? In our first scenario, we are going to drop our cell into this sugar solution with a water potential of negative 0.629. Which way is the water going to move, in or out of the cell? It's going to move out, yeah. We're moving from an area of a higher water potential number to an area of a lower water potential number. Yes, I know, those negatives really mess with you. I was telling this group over here, it took me an embarrassingly long time to get my head wrapped around these negative values and what was higher and what was lower when I first learned about this concept. All right, we have that same cell, but this time we're dropping it into a solution with a water potential of negative 0.174. Which way is the water going to move? Into the cell, yeah. We have a lower water potential number inside of our cell. And so what part of our water potential equation is likely driving the water movement here? Is it the solutes or is it the pressure? Yeah, solutes. Yeah. We really haven't talked too much about pressure yet. It's typically solutes that play a larger role the majority of the time. All right, let's take a break. Let's do another 15-minute break here. I'm gonna go, I heard the doors are unlocked. I'm gonna unlock the doors of the growth chambers here. All right, plants are looking good. Lots of flowers, I like to see that. I was looking at our schedule. We are scheduled to tear down the experiment on Monday after the exam. Which can be a bummer, right? I don't think we're going to have time to measure biomass, right? In order to measure biomass, we have to dry them overnight, and I think if we did that, I'd really be giving you a very small amount of time to write the report, and I don't want to do that too. So I know that in your hypothesis you mentioned biomass. We might not be able to measure it. I'll take a closer look at it, but heads up. Otherwise, anything unusual happening with your plants? I have seen some like fungi in there, lots of algae, keeping that humidity high. Yeah, all right. I mentioned I could smell that humidity when I walked into that room today. All right, we're going to continue our talk on water and how water is moved. I'm thinking likely we'll get out of here a little early today. But we're going to start talking about long distance transport of water within the cell. And let me just put my captions back on. We're still recording. All right. So we talked about diffusion. We talked about osmosis, right? Cell-to-cell movement of water, essentially. That process is really slow, all right? For a typical plant, for water to get from the root cells all the way up to the leaves where they'll eventually evaporate out of the stomata would take centuries, right? It is a very slow process. So there's another process that is being utilized to transport water longer distances, right? And that process is called bulk flow, right? So bulk flow is the movement of these water molecules due to a pressure gradient, right? And so we're not thinking about solute concentrations when we're thinking about this long distance flow, right? Now solute's kind of being put on the back burner a little bit, and pressure is becoming predominantly more what's driving this flow of a lot of water all at once, right? We're thinking about water moving these long distances. So again, when I say long distances, from like roots to leaves, right, spanning the whole plant, right, we're utilizing those vascular cells, right? We're utilizing the xylem, right? And when we look at these pressure gradients, right, like I mentioned, it's kind of like a pull, right? So the water is evaporating out of the leaves, and as it's evaporating, it's pulling the chain of water up through the plant, right? But the whole plant itself, right, there's multiple distances that water has to move, right? First of all, water has to get into the roots, right? That's more of a short distance movement, right? Water has to get through all the cells of the roots and get into the xylem. That's a little bit more of a short distance movement. Once it reaches the xylem, right, now it's going to move via bulk flow throughout the entire plant, right? When it gets to the leaves, it has to leave the xylem, right, and then eventually exit out. So we have a lot of different movement concepts working at the same time here. And we're going to basically follow the order of which plants start at the roots and go, or the water starts at the roots and goes throughout the entire plant here. All right. So if we're thinking about whole plant water transport, right, we're really talking about, movement of water short distance and long distances. But the driving force is transpiration, right? So transpiration is the loss of water through the stomata of the leaves, right? So we know the stomata are those openings in the leaves where gas exchange is occurring, right? Plants are receiving carbon dioxide there. They are losing oxygen there. Well, they're also losing water through the stomata as well, right? Essentially, water is evaporating out of the stomata, right? And so, as water evaporates out, it's pulling the water up through the entire plant, right? Now again, water potential is again one of the driving forces, right? So the water potential of air is really low. It's a really low value, right? And so water wants to go from areas of high to areas of low, right? So water is going to be pulled towards that dry air because of its low water potential, right? So we have this really low water potential of the air. In fact, this is a very great image here that shows you water potentials we might expect. And in the air, we might expect a water potential anywhere from -10 to -100 megapascals. That's really dry, right? That's a very low number. Whereas inside of the leaf, right, the water potential is -7, right? So water is going to want to move from the leaf to the air, and it does so by leaving the stomata through transpiration. All right, so this is like an overview of what we will break down. All right. If we talk about whole plant water transport, we can talk of it as the SPAC, all right? So the soil plant air continuum, right? We're moving throughout the entire plant, from the soil to the plant to the air. Right? So air is drier than the plant, and transpiration is occurring. Right? Like we just mentioned on the previous slide. To get to the leaves where transpiration is happening, right, water has to do this long distance transport up the plant, right? And that's due to bulk flow, right, from the pressure gradients. Right? So there's a difference in pressure here. Right? It's being pulled, it has that negative pressure pulling that water column up throughout the plant. Water has to enter into the roots and flow across the roots. Right? And that's going to be driven by the pressure gradient, but also the solute gradient. And the water actually has to get near the roots before the roots can actually take up that water. And that movement of water in the soil, right, is going to be due to pressure gradients. Right? So very great overview. Right? We're going to start at the roots and work our way up. All right, so starting at the roots, even further out than the roots, we're starting at the soil. All right, so plants take up water from the soil, right? That soil solution surrounding all of our soil particles. If we look at our epidermal cells here, our root hairs projecting out from the main root here, okay? It's taking up the water surrounding all of our particles, and it does so via a pressure gradient. Right? Again, it's a pull. The pressure potential, right, part of our water potential equation, is negative towards the root. It's more negative towards the root. Right? And so because it's negative, it's pulling that water towards the root. Right? And so because it's a pull and we're pulling, we can say it creates this pressure gradient. Right? And that water is going to move via bulk flow towards our root. Right? So we have that pull from the roots. It's actively sucking the water out of the soil. It's creating that pull, creating that negative pressure. Right? And that water is traversing the soil due to a difference in the pressure potential. Right? All right, if we think about how water enters the roots, we often think of, oh, it's the roots. Water is going to enter everywhere in the roots. It's not necessarily the case. If we think about water uptake, first of all, we have to have good contact with the soil. Right? We have to have a lot of surface area in order to explore all the bits and pieces of our soil, get as much water access as we can. Right? We know that root hairs greatly increase that surface area. Right? But the part of the roots that's doing most of the uptake of water is actually the root tips, the younger roots. Right? The older regions actually get what's called, they become superized, so they actually have this waxy layer on them, and they become really poor at taking up water. Right? If we look at our graph that's up here, right, on the bottom we see the distance from the root tip. Right, so we have the growing tip towards the zero axis point here, and then it gets further and further away from the root tip. And we have the rate of water uptake along our vertical axis. And we can see it's this growing tip that takes up most of the water. And the further we get away from that new growing tip, the less water is being being taken up by the water. So it's those newer regions that are really playing more of a crucial role in creating this pull in the soil, this pressure gradient in taking up the water. Right? And the reason is we really want to explore the soil, right? We want to promote new growth, right? And so we're ensuring that these root tips are growing and exploring the soil. All right, so water has been pulled towards the roots via this pressure potential. Right? Now the water has to enter into the roots. Right? The water is actually going to enter into our root cells due to a difference in the overall water potential. Right? So now we're taking solute and pressure potential. We're adding it together to get our overall water potential. Right? And it's going to be that difference in water potential from outside of the roots and inside of the roots that's going to cause the water to actually move into the root cells themselves. Right? And remember, we're going from areas of high to areas of low. Right? So the water in the soil has a very high water potential. These cells have a lower water potential, and that is driving that water inwards. Right? This is how water actually enters into our plant. Right? So negative pressure pulling that water towards our root, water potential causing the water to enter into the root. All right, once it gets into the root, right, it has to reach our vascular tissue. And if you remember, our vascular tissue, specifically that's our water conducting cells, are the xylem, right? The xylem provides the longest pathway of movement for water, right? And this is where we're moving due to bulk flow, due to these pressure gradients, that pull of water up the water column, right? Now the xylem, right, we talked about in broad terms, xylem are our water conducting cells. There's actually different types of xylem, right? One of them would be tracheids. They're more long, they're skinnier. The other are these vessel elements. They're shorter, more stout, but wider, right? But no matter the type of xylem cells, right, they're all going to have these plates on their ends that allow for that direct movement from cell to cell to cell. Now I know this isn't on the slide here, but our xylem cells, they're actually dead, right? We talked about phloem cells being alive, but they lack a lot of organelles. Our xylem cells are dead when they're mature, right? When they're actually functioning. They don't have any organelles on the inside. They don't have a plasma membrane, right? This allows for very quick movement of water through these cells. All right, so water has now entered into the xylem of our plant cells at the roots. We have to get this water from the roots to the leaves, right? And this process is called cohesion tension theory, right? So we have cohesion, so water molecules like to stick together, right? And we have some tension or some pull, right? Some negative pressure causing the water to move. So that negative tension, right, is created at the top of the tree by transpiration, the water leaving the leaves. Right? So the water has left the leaves. It's pulling out new water as it leaves. Right? But water is sticking together because they're cohesive to one another. Right? So if you remember in that Amoeba Sisters video, they said a good analogy was like beads on a string. Right? They just go up in a chain. They like to stick together. So it's the transpiration that causes the tension, and it's the cohesion between molecules that cause those water molecules to move up with that tension that's created. Right? The cohesion tension theory. Pause for a second here. See a lot of us still writing. All right, I mentioned this so many times that I made it a very quick thought question because it's so important. All right, but maybe a minute for you to digest the information so far and answer the question, does water move in the xylem via positive or negative pressure? Is it a push out of the plants or is it a pull out of the plants? feel like we've all come to a conclusion. All right. Is it positive or is it negative pressure? Negative, yeah. So negative pressure is a pull, right? We're pulling water out of the plant as it evaporates from the leaves. Are there any questions? We're about to dive in just a little bit deeper into this. Alrighty. Alright, so the tension is causing the water to be pulled out of the plant. But how is this tension being created? Right? Well, it's being created via transpiration, right? So if we look at a cross section here, right, we see water, this is our vascular bundle here, is leaving our xylem, going into the surrounding cells, right? But because the air is so much drier outside of the plant, the water tends to leave our cells, right? They turn into water vapor, right, and evaporate out of our stomata. Right? So we have our water, it's leaving our cells, it actually kind of forms this like film on the outside of cells. Because again, water likes to stick together, right? But water really doesn't like to change forms. It has this high specific heat, right? It doesn't like to turn into vapor. But because this air is so dry, right, it's such a massive pull on this water, right, that water will leave this film, right, turn into water vapor, and exit out of our stomatas. Right? Now remember, all the while, all this water is still connected to all of that water being pulled up the plant. So as some water molecules vaporize and leave the plants, others are going to take its place. Again, just kind of emphasizing this point here, right? Water is pulled into those mesophyll cells. Some of it's used for photosynthesis, right? But some of it is going to exit the leaves because the air is really dry, right? Now, the plant doesn't necessarily want the water to leave, right? But that pull, that gradient, that water potential gradient between the leaves and the air is so strong that it happens, right? So the plant needs to bring the water up to the leaves in order for photosynthesis to occur, right? We're using that water, creating that oxygen. But because the water is in the leaves and the stomata are open, there's such a pull that the water will leave the stomata as well, right? Now, if we think about it, right, the driving force is this gradient in water potential, right? Or we could think of it as like a water vapor concentration gradient. There's less water vapor in the air than there is in the leaves, right? There's a lower water potential in the air than there is in the leaves, right? And the reason our air is so dry, right, we have the sun, right? The sun is warming up the atmosphere, making it dry out there, right? So we could say that the driving force here, right, is the sun. That water transport is solar powered, right? But like I mentioned, plants don't necessarily want to lose that water, right? It's just a byproduct of the stomata being open and those leaves needing the water for photosynthesis, right? Some of that water is going to evaporate and leave the plant, right? Now the plant doesn't want this to happen out of control, right? If there was nothing stopping water from leaving the plant would run out of water and die, right? It wouldn't have enough water, right? So there's a couple of different ways that plants have to regulate how much of this water is going to be lost, right? One of which is this boundary layer resistance, right? I'll elaborate on it on the next slide as well, right? Basically, there's a layer of air above the leaf, very, very tiny layer, right? Microscopic layer of air that's a little bit more humid than the outside air itself, which stops the water from leaving so fast. And then we have the stomatal resistance, which again I'll elaborate on as well. Right? And that's simply the resistance of leaving the plant, right? Whether the stomata are open or closed or how much they are open. Right? So I'm going to elaborate on this boundary layer resistance first. Right, same definition as on this page. Boundary layer resistance, right? Like I said, is this resistance due to an unstirred layer of air through the leaves, right? And I'm actually going to use the blackboard for this. So if we have our leaf here, we have all of our palisade mesophyll cells, right? Maybe we'll say this is where we have our stomata, right? Our gap here, right? There's a layer of unstirred air, basically like this really humid air, just outside of the leaf, right? So that kind of lowers the difference in water potential between outside of the plant and inside of the plant, right? So the air might be really dry. It might be like negative 100, right? Inside of the plant, it's like negative 7 megapascals, right? If there's just this difference here, water would leave pretty fast. But if we have this layer of air that's very humid, maybe this layer of air is, we'll say it's like negative 10. I'm not exactly sure what the water potential of this boundary layer is, but it's something in between, right? Now we're just having to deal with negative 7 to negative 10. That means water is going to leave, but it's going to leave a little less quickly, right? It has to go through this very humid air first, right? It kind of slows down the rate of evaporation, right? And so in order to have these boundary layers, right, plants have all these different adaptations to increase the size of this layer, right? So they might need to increase the size of this layer so it's, even thicker, even harder for water to leave, right? Leaf size can influence this, right? We look at these really small leaves here, right? They tend to have this lower boundary area because there's just not as much leaf, right? So they can't build up that layer as well, that humid layer. But also the shape of the leaves. We again look at this leaf here, right? It's kind of curled inwards, right? So it's kind of like this pocket that this plant is creating, which can increase the humidity inside of that pocket. Right? But also environmental factors like wind speed, right? If there's a brisk wind out there, right, it's going to brush away this boundary layer. It's going to blow away that boundary layer much more quickly, right? Rather than if there's no wind at all, that boundary layer is going to be undisturbed, right? So plants have found ways to increase the boundary layer to reduce the amount of water loss, right? That's the ultimate goal here, reducing the amount of water that is lost through the stomata. All right, so boundary layer, right? This can also be influenced by like trichomes, so the hairs on leaves. The hairs can also trap in more of that humid air as well. But we also have stomatal resistance, right? The actual resistance associated with moving through the stomatal pore. Right? This one's fairly easy to understand compared to boundary layer because it's really looking at, is the stomata open? Is the stomata closed? Right? If the stomata is closed, obviously air is not going to be able to exit the plant, right? As well as the water vapor. Right? If it's open, right? And maybe it's wide open rather than semi-open, right? Water is going to leave much more quickly. It has a larger pore to diffuse through. Right? So it's whether or not those stomata are open and closed, or how much they are open. Right? Now, plants have biological control over this. Right? So unlike wind speed and boundary layer, right, where plants can have some adaptations to maybe increase their boundary layer, maybe they have those hairs, right? They're not really going to be able to control the wind speed, right? But plants can control whether or not their stomata are open and closed, right? There's different signaling pathways going on here, right? If it's very dry, they can close their stomata, right? But it's also a trade-off. If you close your stomata, you're also not getting gases inside the leaf, so you can't photosynthesize, right? So they have to have this trade-off between how much water they're willing to let go out of their leaves, right, versus how much photosynthesis they can actually perform. And there's a lot of different ways that plants regulate that. We won't go into all those details. Right? All right, so if we summarize water movement throughout the entire plant, right? It's kind of a review here. Right? We look at the soil, right? Water is being driven towards the soil due to a difference in pressure. Right? The roots are creating this pressure, a pull of the water towards the roots. Right? To actually enter into the roots, it's caused by a difference in overall water potential. Right? So we're getting into that xylem, right? Entering the roots, moving across the roots, entering into the xylem is caused by a difference in that water potential. Once it reaches the xylem, right, it's basically this highway system here. We can move water very quickly, right? And water is driven by the pressure gradient, right? So the pull, right? And that pull is generated by the leaves, right? Water evaporating out of the leaves due to water leaving the leaves because the air is very dry. Okay. All right, so transpiration is the driving force behind all the water movement. If there wasn't any transpiration, right, water wouldn't even be taken up by the roots in the soil. Right? We have to have that pull basically for this whole system to work. All right, I wanted to end this lecture on how we can actually measure these types of things, right? So we can measure the gas exchange occurring out of stomata, right? We can measure how much of stomata is open and closed, right? So basically we take this machine, right? We clamp it onto a leaf, right? And this is me doing some of my thesis research out of California here. We clamp it onto a leaf, right? Inside this chamber that's clamped onto the leaf, we know precisely the amount of carbon dioxide and oxygen, right? And we monitor those levels to see how they change, right? If carbon dioxide decreases, we know the plant is using it, right, for photosynthesis, right? And so when we do these types of studies, we can infer their photosynthetic rate. So how much are they photosynthesizing? We can infer their transpiration rates. Right? So that chamber also measures the amount of water vapor that's being released, right? Which can tell us how much this plant is transpiring. We can see what time of day these plants are active. So some plants will not photosynthesize during the day, right? Some plants have these different adaptations that they will actually do a lot of their photosynthesizing during the night, right? They'll capture energy from the light during the day, store it, and then actually make the sugars at night. This can tell us something about the stress level of plants, right? So really stressed plants are not going to transpire as much. They're not going to have these high photosynthetic rates. But we can also look at something like water use efficiency. So how efficient are they in using their water? Right? So water use efficiency is the ratio of assimilated carbon dioxide. So how much carbon dioxide is coming into the plant and coming into the Calvin cycle and being made into sugars, right, versus the amount of water loss. So how efficient are they with their photosynthesizing and their transpiration? Right? And this can really help us to understand, how efficient plants are, right? Some plants are more efficient at this than others just due to their evolutionary history, right, where they evolved. Desert plants tend to be pretty efficient at this because they have to be. Right? They don't get a lot of water. But one of the statistics that I like to share that is pretty incredible is that, in general, plants lose about 500 molecules of water per 1 molecule of CO2 used in photosynthesis. That's crazy. That's a ton, right? For just 1 molecule of CO2. So it's a pretty cool statistic, right? And there are ways that we can study these. Right. If we think about transpiration happening in space, right, and does transpiration happen normally? Right. Well, there's been some studies out there, right? Some studies are ongoing, right? So we could have like the effect of gravity on the transpiration in plant leaves here, right? That's been a study that's been done. It was done in 2009, and they basically found that the less gravity there was, the less transpiration, right? And so I'm hypothesizing here that it must be something to do with there's just less pull of water out of the plant due to there being less gravity, right? Right. Another study here done in 2016, right, said that transpiration, yes, was affected, right, but they have developed technologies to kind of reduce the amount of transpiration that would be affected. You've seen that greenhouse when she was watering those plants. It had those sides of plastic trying to keep it very humid inside there, right? But also they have lights to encourage photosynthesizing, which means plants have to open their stomata, right? They have to transpire, right? But there's also been a study, 2005, right, which is a little bit of later of a study, right, that microgravity really doesn't alter gas exchange, right? But they admitted in the study that there's more to explore. There's more that needs to be confirmed, right? I think it would be really cool to bring one of those machines up to the ISS and clamp it onto leaves to see what's going on. But to my knowledge, that has not been done yet. I imagine the machine is not going to work as well in microgravity, and there's technology that we have to develop in order for that to happen. So we know a little bit about how it works in space. Again, still ongoing because space is just such a harsh environment for a lot of scientific studies. All right. We're going to end a little early here. So, for our homework, I have a couple of videos here. Gravity lab report. I think I said it was due today. It's due tomorrow, right? That's why all of you were looking at me kind of freaked out. I thought maybe it was because you didn't know what you were doing in your report, but it's because I missaid the due date. It is due tomorrow. The annotated bibliography, that was due today. Well, is due today. I think I have it set at midnight. Right? I'm a little lenient on if you get to me by 2 a.m., right? How to Grow a Planet assignment is due tomorrow, so hopefully you're working through that. Right? Exam 2 on Monday. Tomorrow will be a short lecture and then a fun day in the greenhouse. And then, of course, our last discussion next Tuesday. Any questions? All right, you'll notice that the next slide here is linked to video. It's a really great overview. I'm not going to show it in class, but I put it on the slides in case you wanted another visual to kind of link all these concepts together.
Put in